Good evening, ladies and gentlemen. After 30 days of successful program, science program, we are at the end of the tunnel in the sense, 31st day, the valedictory address day. You know that the Bangalore Science Forum was started in 1962. Of course, you remember Dr. Maya, you are the person responsible for the starting of the Bangalore Science Forum. It's better if I give you one or two things about it. Then, Dr. Maya was professor of physics in National College, and College Science Association is to conduct two functions. One is inaugural address in the month of June, other is the valedictory in the month of December. Only two science functions throughout the year. Then Narasimha thought there's no meaning in conducting only two science functions. Science functions should be there all the way throughout the year. Then we formed the Bangalore Science Forum. And Ramakrishna because Ramakrishna and myself were the students, and he started the forum. They used to make fun of Narasimha and also fun of the young students like us. What will they do? Science forum would remain even for 1 year or 2 years. It's very difficult to conduct lectures. But just imagine 62 that day. Today it is 2015. You see the number of audience for science lecture. I think that Narasimha's stand is clearly vindicated, actually. I am continuously doing. I don't think we have let off a single Wednesday since 1962. Then in 1978, of course Steve Jobs was not there. Of late, Steve Jobs has said, we were 4 or 5 years back, if you do not know music, you are more dead than alive. But then, in 78, the music, the people, and Dr. Simha said the music is very important, even more important than science. Then he used to call me, Bani Hona Legated, Amma City Institute here. And I used to follow him always in my life, 25 years difference between us, Narasimha and me. And then in the evening program in the City Institute, suddenly he got an idea that Ganesha festival Ganesha festival is there. Sankranti festival Deepavali festival music festival yes, why can't we do a festival for science, he said. I think the World Science Festival was coined in a music concert in 1978, maybe in the month of June, Then he started in 1978, 15 lectures, 79, 30 lectures. So difficult to fix 30 lectures. 80 monitro, success begets success, 85, 1995, 2000, 2010, 2015 believe ನಾನು ಏನು ಆಸೆ ಅಂತ ಹೇಳಿದ್ರೆ ಇದು 3000 ಇರುತ್ತೆ ನಾನೇ ವೆಲ್ಕಮ್ ಸ್ಪೀಚ್ ಮಾಡೋದು ಆಸೆ ಇದೆ ಸೈನ್ಸ್ ಫೆಸ್ಟಿವಲ್ July 1st is fixed, July 31st is fixed, year is not fixed. 38 years ಇದು ಮಾಡಬೇಕಾದ್ರೆ ನ್ಯಾಚುರಲ್ ಆಗಿ it is due to the complete cooperation of all the professors at different institutions of the Bangalore and also other places, and audience like you, that's also important, every one of them have attended these lectures. ಹಾಗಾಗಿ ಇವತ್ತಿನ ದಿನ we are at about 2665 ಪ್ರಿಂಟ್ ಆಗಿದೆ, 66 ಬೇಕಿದು, ಈಗ ಒಂದು ಲೆಕ್ಚರ್ ಎಕ್ಸ್ಟ್ರಾ ಇದೆ, 2666 lecture. In a short thing, in a few minutes, Dr. B. Balaram, former director of Indian Science, will deliver this valedictory address on chemical ecology, the discipline bridging chemistry and biology. Today, on behalf of the Bangalore Science Forum, on behalf of you and on behalf of me, I extend a very cordial welcome to Dr. B. Balaram. Balaram, if I am correct, 1977 ನಲ್ಲಿ ಇನ್ಸ್ಟಿಟ್ಯೂಟ್ ಬಂದಲ್ಲ, ಅವತ್ತಿಂದ ಇವತ್ತವರೆಗೂ more than 50 lectures ಬಿಟ್ಟಿದ್ದಾರೆ. ಒಂದು ನಾನು ವೆರಿ ಹಂಬಲ್ ಪರ್ಸನ್, ಬಟ್ ಒಂದು ಹೇಳ್ತೇನೆ ನಾನು, ಒಂದು 5-6 years back at about 5:30 AM ಡೆಕ್ಕನ್ನಲ್ಲಿ ಡಾಕ್ಟರ್ ಬಲರಾಮ್ ಅಪಾಯಿಂಟೆಡ್ ಆಸ್ ಇಂಡಿಯನ್ ಸೊಸೈಟಿ ಡೈರೆಕ್ಟರ್ ಅಂತ ಇತ್ತು. ನನ್ಗೆ ಎಷ್ಟು ಸಂತೋಷ ಆಯ್ತು ಎಕ್ಸ್ಪ್ಲೈನ್ ಮಾಡೋಕ್ಕಾಗಲ್ಲ. ಸಿನ್ಸ್ ಐ ನೋ ಇಮ್ ವೆರಿ ವೆಲ್, 5:45 ಫೋನ್ ಮಾಡಿದೆ ಅವ್ರಿಗೆ. ಸೊ ಕಂಗ್ರಾಚುಲೇಷನ್. ಅವ್ರಿಗೆ ಎಷ್ಟು ಇದು ಅಂತ ಹೇಳಿದ್ರೆ ನಾನು ಕಂಗ್ರಾಚುಲೇಟ್ ಮಾಡಿದ್ದಾರ ತಕ್ಷಣ ಏನಂದ್ರೆ, ರಾಮರಾವ್, ಟುಡೇ ಐ ಹ್ಯಾವ್ ಅಗ್ರೀಡ್ ಟು ಡೆಲಿವರ್ ಎ ಲೆಕ್ಚರ್ ಇನ್ ಬ್ಯಾಂಗ್ಳೂರ್ ಸೈನ್ಸ್ ಫಾರ್ಮ್ ಅಟ್ ಅಬೌಟ್ 9 o'clock. ಅದು ಕಮ್ ಇಯರ್ ಅಂತ ಹೇಳಿದ್ರು. ಮುಂದೆ ಇನ್ನೂ ಚಪ್ಪಾಳೆ ಕೊಡ್ಬೇಕಾದ್ರೆ ಅಷ್ಟೇ ಎಲ್ಲ ಹೇಳ್ತೇನೆ. ಆ ಸಮ್ಮರ್ ಸ್ಕೂಲ್ ಯಾವ್ದಂದ್ರೆ SLC ಸಮ್ಮರ್ ಸ್ಕೂಲ್, ಇಲ್ಲಿ ಬಂದು ನಿಂತ್ಕೊಂಡ್ರು. ಇನ್ನ ಡೈರೆಕ್ಟರ್ ಅಪಾಯಿಂಟ್ ಆಗಿದ್ರೆ ಐ ಆಮ್ ಎಕ್ಸ್ಟ್ರೀಮ್ಲಿ ಬಿಸಿ, ಐ ಹ್ಯಾವ್ ಟು ಗೋ ಟು ಡೆಲ್ಲಿ ಅಂಡ್ ಅದರ್ ಪ್ಲೇಸ್, ಡೋಂಟ್ ಟಾಕ್ ಟು ಮಿ ಫಾರ್ ಅಬೌಟ್ ಅ ಮಂತ್ ಅಂತ ಹೇಳಾಯ್ತು. ಇವ್ರು ಹಾಯಾಗಿ ಬಂದ್ರು 9 o'clock. ಆ ಹುಡುಗರು ಏನ್ ಹೇಳಿದ್ರೆ, As the Director of the Institute of Science, this is my first public appearance. And now, Yava, man of very few words. Now, I had to speak about 5 words and 1 full sentence. His answer is yes only, no Iloila. He had never refused anything to National College. Actually. And Dr. Balaram received his bachelor degree in chemistry from Ferguson College, University of Pune, followed by a master degree from the University of Science and Technology, Kanpur, and his PhD degree from Carnegie Mellon University with Axel A. Fortner grade. After a postdoctoral stint at Harvard University with Nobel laureate Robert Burns Woodward, he returned to the Institute of Science, where he has been ever since as a faculty member in a molecular biophysics unit. During his PhD, Balaram studied the use of negative nuclear Overhauser effect signals as proofs of micromolecular conformations. As a postdoc with Woodward, Balaram worked on the synthesis of antibiotic erythromycin. The agricultural erythromycin is such a charged drug for the doctor now. He has worked in that field. And his main area of research has been the investigation of the structure, conformation, and biological activity of designed and natural peptides. To do this, he has extensively used techniques such as nuclear magnetic resonance spectroscopy, infrared spectroscopy, and circular dichroism along with X-ray crystallography. He has been a major contributor to the evolution of factors influencing the folding and conformations of designed peptides, and has investigated structural elements playing a key role in the formation of secondary structural motifs such as helices, beta-turns, and sheets. Along with Isabella Cawley, a frequent collaborator, he has also pioneered the use of alpha-amino isobutyric acid to induce and retain helicity and constrained peptide conformations. He has authored more than 400 research papers and is a fellow of the Indian National Science Academy. In fact, the most interesting part is he was also a colleague of late Professor G.N. Ramchandran at the Molecular Biophysics Unit of the University of Science. Without G.N. Ramchandran thought, the biochemistry or biotechnology won't start at all. Naturally, he has got a large number of awards. INSA Medal for Young Scientists, UGC Career Award, UGC National Lecturer, SS Bhatnagar Award, Alumni Award for Excellence in Research by University of Science, DAE C.V. Raman Lecturer, G.D. Birla Award, Cross Award, Jawaharlal Nehru Birth Centennial Lecturer, and Jawaharlal Nehru Birth Centennial Visiting Fellowship of INSA. He is a Fellow of the Indian Academy of Sciences, Bangalore, and the Academy of Sciences for the Developing World. Yavudu bittilla actually. Nanu kelidare nanu insurance system. But finally, all these are gapped by 2 awards. One is Padma Shri earlier stages. In 2014, he got Padma Bhushan from Government of India. He is there in the Institute of Science as a lecturer since 1973 and director for about 9 years, 2005 to 2014, and current science editor, Chem Biochem, European Journal of Chemical Biology, Editorial Advisory Board, Biopolymers, Protein Engineering, Design and Section. Antari ಇವತ್ತಿನ ಬರೆದಿದ್ದಾರೆ ನಮ್ಗೆ ತುಂಬಾ ಸಂತೋಷ. ಅದಲ್ಲದೆ ಇವತ್ತು ಮುಗಿತಾರು ಸಹ we are not keeping quiet. Next month program is already ready and in print tomorrow you get it. ಅದೇನಾಯ್ತು ಅಂತ ಹೇಳಿದ್ರೆ ನಾನೆಲ್ಲ ಮಾಡಿದ್ದೆ. CO ಮಾರ್ಕಷ್ಟೊಂದಿಗೆ ಸೊ ಮಾಡ್ಕೋಬೇಕು ಯಾವುದೋ ಒಂದು ಸಬ್ಜೆಕ್ಟ್. ಆಮೇಲೆ 3:30 ಅವ್ರು ಫೋನ್ ಮಾಡ್ಕೊಂಡು ಸಬ್ಜೆಕ್ಟ್ ಇದು ಮಾಡಿದೆ. Of course, first Wednesday we will have the film show on science subject because we have this one film show, and then 12/8 Mr. H.N. Bhargava, ex-mission director ISRO, will speak on network-centric warfare. Then 19th Wednesday, Mr. D.A. Mohan, retired EDO of BL and CEO of Defense Aerospace Computing International, advances in defense electronics. Then Professor R. Chandrasekhar, professor from University of Information Technology, will speak on trends and challenges in online education. Those subjects of that, when it comes to big data analytics. ಇಷ್ಟು ಒಳ್ಳೆ ಸಬ್ಜೆಕ್ಟ್ಸ್ ಇದೆ, ದಯವಿಟ್ಟು ಎಲ್ಲರೂ ಬರಬೇಕು. ಬಟ್ ಇನ್ನೊಂದು ನಿಮ್ಮನ್ನೆಲ್ಲ ವೆಲ್ಕಮ್ ಮಾಡಿದ್ರಲ್ಲೇ ನಿಮ್ಗೆ ತುಂಬಾ ಥ್ಯಾಂಕ್ ಮಾಡ್ತೇನೆ. ನೀವು ಬರೆದ್ರೆ ಯಾವ ಪ್ರೋಗ್ರಾಮ್ ಮಾಡೋಕ್ಕಾಗೋದಿಲ್ಲ. ಐ ನೋ ದಟ್ ವೆರಿ ವೆಲ್. ಬಟ್ ಇದಾದ್ಮೇಲೆ ದಿನ ಇಲ್ಲಿ ಬರ್ತಾ ಇದ್ದೀರಾ, ಮಧ್ಯ ಕಾಫಿ ತಿನ್ನು ತಗೊಂತಾ ಇದ್ದೀರಾ. ಒಂದಸ್ತಿ ಹ್ಯಾಬಿಟ್ ಟೇಕ್ಸ್ ಹಾರ್ಡ್ ಅಂತ ಹೇಳ್ತಾರೆ. 31 ದಿನ ಆಗೋಗಿದೆ. ನಾಳೆನೂ ಬಂದ್ಬಿಡುವ ಎಷ್ಟು ಮತ್ತೆ ಏನು ಬರಿ ಮಾಡೋಕ್ಕಾಗ್ತಲ್ಲ. ಫಸ್ಟ್ ಕ್ಲಾಸ್ ಇಸ್ರೋ ಅಂಡ್ ಅದರ್ ಪ್ಲೇಸಸ್. ಇನ್ಫ್ಯಾಕ್ಟ್ ಇಸ್ರೋ ಅವರಿಗೆಲ್ಲ ಎಷ್ಟೊಂದು ಥ್ಯಾಂಕ್ಫುಲ್ ಇರಬೇಕು ಅಂತ ಇದ್ದೆ. ಇನಾಗ್ರೇಟ್ ಮಾಡಿದ್ರೆ ನಮ್ಮ ಸ್ಟೂಡೆಂಟ್ ಡಾಕ್ಟರ್ ಕಿರಣ್ ಕುಮಾರ್ ಅವರು ಅವರೆಲ್ಲ ಚೆನ್ನಾಗಿ ಕೋಆಪರೇಟ್ ಮಾಡಿದ್ದಾರೆ. ಅಂಡ್ ಇನ್ಫ್ಯಾಕ್ಟ್ ಡಾಕ್ಟರ್ ಬಲರಾಮ್ ಅವ್ರಿಗೆ ಜ್ಞಾಪಿಸ್ಕೊಂಡ್ರೆ ನಮ್ಮ ಅದೃಷ್ಟ ಅನ್ಸುತ್ತೆ. ಅದು ನನ್ನ ಮನಸ್ಸಿಗೆ ಹೇಳಿದ್ರೆ ಡಾಕ್ಟರ್ ಬಲರಾಮ್ ಅಂತ ಇನ್ನೂ ನಾನು ಮಟ್ಬಿಟ್ಟೆ. It is because of the efforts and courtesy of Dr. Balaram, I got Nobel laureate Dr. Nigeshi, Nobel laureate Bruce Hoffman, ಅದಲ್ಲದೆ ಇನ್ನೊಂದು ನೇಮ್ ಹೇಳಿದ ವುಡ್ ಬಿ ನೋಬಲ್ ಲಾರೆಟ್ ಇಂಟ್ರೊಡ್ಯೂಸ್ ಮಾಡ್ಬಿಡ್ತಾರೆ ಅಷ್ಟು ಇದು ಮಾಡಿದಾಗ ನನಗನ್ಸುತ್ತೆ ಇನ್ನ ಗ್ರೇಟ್ ಸೈಂಟಿಸ್ಟ್ ಇದ್ದಾಗ ನಾವು ಸಹ ಅವ್ರ ಜೊತೆ ಬದುಕಿದ್ದೀವಲ್ಲ ಇಂದ ಏಜ್ ಅಂತ ಹೇಳಿ ನನಗೆ ತುಂಬಾ ಸಂತೋಷ ರಿಕ್ವೆಸ್ಟ್ ಡಾಕ್ಟರ್ ಬಲರಾಮ್.
I'd like to thank Dr. Balaram for inviting me to the Science Forum once again. I've been coming every year to the Science Forum for so many years. I hope that this will also be my validation. Because I think you should find people who are younger to come and talk to you about the new emerging disciplines of science. But what I thought I would do is really talk to you today about a subject which you will hear about more in the future, a subject called chemical ecology. The reason I'm interested in chemical ecology is that institutions in Bangalore, both the Indian Institute of Science as well as the National Center for Biological Sciences of the Tata Institute of Fundamental Research, are now starting programs in chemical ecology, and there will also be a national program which links the universities and institutions in the northeastern states of India with Bangalore as a Bangalore Northeast program to further research in this area. Now there are two words in the subject chemical ecology, two subjects, chemistry and ecology. Now of course, if you ask, I was trained as a chemist, but if you ask me to define chemistry, it will take me a little while. If you ask me to define ecology, I would find it difficult. So I did what all of you would possibly do, go and consult Wikipedia. And the moment you do that, you get a definition which somebody has thought about and put onto the internet. And of course, chemistry is a branch of the physical sciences that studies the composition, structure, properties, and changes of matter. So in chemistry, we talk about molecules, we talk about the way molecules are, atoms and molecules are connected to one another. We talk about the reactivity of molecules, where one molecule is converted to another. Those are the transformations of matter. So one talks about dynamics of molecules, structures of molecules, reactivity of molecules. Ecology is the scientific analysis and study of interactions among organisms and their environment. You can think about this because a subject like chemistry, sometimes one has difficulty if one has not studied it before. Although I believe that chemistry is the most important subject for everybody to know a little bit about, and you will see why when I go to my next slide. But I will tell you the most compelling reason for you to be friendly with chemicals is simply because you are composed completely of chemicals. And therefore, if you want to understand yourself, there is probably a very good reason that you must understand a little bit of chemistry. There is nothing around you that is not a chemical. So, using the word chemical as some kind of bad word to describe a pollutant or a toxic substance is incorrect, because everything, in fact, in nature is chemical in nature. So chemistry pervades all of nature. How do organisms interact with one another? Now, if you are going to interact with your neighbors, with your family, with your friends, with your enemies, you interact with them primarily by speaking to them. So what we are really conveying information, we are conveying information through sound. And therefore we largely use sound waves, transduce sound waves into information that we can distill and understand. All of it is undoubtedly a very mysterious process, the process by which we see and we understand. We see the world around us, that's the sight of vision, and of course the ways in which we transduce sound into a meaning that they are able to understand. But organisms in biology generally do not talk to one another. And since they do not talk to one another, you have to ask the question, how do all organisms communicate with one another? Because they are in fact communicating with one another. Ecology largely requires field biology. That means you go out into the field, you go out into the forest, you go out and study nature. The most dramatic study of nature throughout the history of science is the study of nature which Charles Darwin carried out when he went on the voyage of the Beagle. He observed nature wherever he went, and it was by observing nature wherever he went that he came to the idea of the unity of biology. The theory of natural selection, evolution, Darwinian evolution, today really unifies biology. Chemistry, the branch of chemistry which is most important for chemical ecology is natural products chemistry. This is to understand all the chemicals that are present in nature. The chemicals produced by plants, the chemicals produced by animals, and very often the chemicals produced by microorganisms whom you can't see them, but Earth is, the greatest population on Earth is of microorganisms. In fact, the number of microorganisms which are living in your gut today is incredible. And these organisms are producing chemicals all the time. One of the most disturbing things is even our moods and behavior might sometimes be controlled by our gut microorganisms. So although you can see them under the microscope, you can't see them with the naked eye, therefore we don't think about them most of the time. But they are producing chemicals of all kinds. Now, of course, to attract your attention to the subject of chemicals, I can't do better than to start with the most recent chemical controversy which has come in the newspapers. That is the case of the Maggie noodles. Now, what did that analytical lab somewhere, I think, in the north of India, discover in Maggie noodles? They found two substances. One substance was lead, and the other substance was what is called monosodium glutamate. And monosodium glutamate is a substance which is viewed with alarm by everybody. It's viewed with alarm by policymakers, by politicians, by doctors. It's just viewed with alarm by everybody. That is monosodium glutamate. Nice white crystalline powder, as most of the other amino acids are. It is sold by the Japanese company Ajinomoto, which is the world's largest producer of amino acids. But there is only one product which carries the name of the company itself. It is called Ajinomoto. So when we talk about Ajinomoto, we are talking not only about a company, but we are talking about this substance, monosodium glutamate. Why do the Japanese use Ajinomoto? They add this substance because it gives a good taste to the food that you have. And therefore everybody's been mixing it with food. It is there in all kinds of foods. It's there in Coca-Cola. And obviously it is there also to some extent in Maggie noodles. Why is monosodium glutamate such a bad chemical? I'll come to that the next slide. But that's the Maggie noodles, that's monosodium glutamate, that's the way Ajinomoto sells it. That is the substance monosodium glutamate. This is the substance glutamic acid. Glutamic acid is a constituent of proteins. It is an amino acid which is present in every protein that we have in the body. It is there in every protein meal that you consume. What is the difference? Even if you know a little bit of chemistry, you will see here is a hydrogen, here there is a sodium ion. What has happened? The amino acid has been dissolved in water, it ionizes. Just like when you put sodium chloride or common salt in water, it becomes sodium plus and chloride minus. This has become glutamate minus and sodium plus. So there is effectively no difference between monosodium glutamate and glutamic acid. But we already have glutamic acid in very large amounts. There's a study, for example, published about 20 years ago, which concluded that the present study led to the conclusion that the Chinese restaurant syndrome is an anecdote applied to a variety of postprandial illnesses. Of course, if you eat too much, there will be a large number of illnesses. All of them cannot be traced to monosodium glutamate. I understood that there is a social science forum also that is going to be started. Here you can see a Japanese product has been labeled in America as causing Chinese restaurant syndrome. I don't think the Chinese have it so much. There's a huge sociological difference between the Chinese and the Japanese. The last thing that the Japanese would like to have is to be confused with the Chinese, and the Chinese don't want to be confused with the Japanese. Glutamate is what is called an excitatory neurotransmitter. What is a neurotransmitter? All the cells in your brain, which look like this, they are separated by a small gap. This is almost like the Cork Strait which separates India from Sri Lanka. A tiny band of water. And across this band of water, if you want to communicate, one cell has to secrete chemicals, and those chemicals have to be recognized by the neighboring cell, and therefore what you have is chemical transmission of information. One thing you must remember if you're in Bangalore. In Bangalore, there is one phrase which one hears all the time, and that is the word information technology. Now, nobody really knows what is information technology. Information technology means information being communicated from one place to the other, various technological means of actually communicating this information. And we've been communicating information all the time without any technology whatsoever. The only technology that we have at our disposal is a technology already given to us by nature. When you think about communication, remember that biological communication is the most remarkable of all forms of communication. Have a baby, starts to talk. The first time the baby talks, the entire family has gathered around, from great-grandparents downwards. Everybody is fascinated by this. Afterwards, we take it for granted because everybody talks practically, with very, very few exceptions. How did this happen? How do we understand? We are already communicating information all the time. But cells communicate with one another through chemicals. So it's chemical signals which get converted into electrical signals, which then get converted into chemical signals again, and that's what happens when the brain is processing information. And the brain today processes information much better than any available computer that exists. It's a soft system. You don't understand how it works. Nevertheless, it works very well. Glutamate now is considered to be the major mediator of excitatory signals in the mammalian central nervous system. Therefore, it's very, very important for communication in the brain. But remember that the neurons of brain cells communicate with one another via secreted chemicals. Now, we will talk about chemical communication in biology. How do cells communicate with one another? See, your body itself is made up of so many cells. Thousands upon thousands upon thousands of cells. The communication between cells is usually, there is a chemical effector. Cell-cell communication usually happens by means of hormones. All of you know the word hormone. All the sportsmen also know the word hormone. So among the most abundantly misused chemicals are hormones in the field of sports. Few days ago, in fact you can learn more science by reading the sports pages than you can by reading any other page. Because if you read any other page in the newspaper, you will only learn about politics and crime. On the other hand, if you read about sports pages, if you read the sports pages, you will be entertained. In addition, you will learn a little bit of science. A few days ago, there was this story about this young lady who had now been cleared now to participate again in the women's events. What was her problem? Her problem clearly was a hormonal problem. But when it is a hormonal problem, where, for example, testosterone, a male hormone, is now being produced in slightly larger amounts in a female body, that might be a natural aberration. On the other hand, if you take testosterone intravenously, it might boost your performance for a little while. That is doping. But to make this distinction between doping, which is banned, and a natural phenomenon over which the person has no control whatsoever is a problem really of science. And therefore, one must remember that hormones are in fact really what determines not only what we are, but the ways in which we interact, the ways in which we behave, and cells are communicating by this mechanism all the time. When we react to pain, for instance, sometimes we are able to bear the most intense pain in an accident. We don't even know how we do it. We do it because the brain has already secreted hormones, neurohormones, which are now in fact very responsible for suppressing the problem of pain momentarily. But how do organisms communicate with one another? So there are two kinds of communication in biology. Cell to cell, your brain to your stomach. After all, you get a signal saying you're feeling hungry, or you get another signal saying you're full, don't eat anymore. Before I came in, Dr. Balaram persuaded me to eat much more than what I possibly could. And eventually you do get a signal which says stop, no more. That's the hormone. Organism to organism. How do these bugs know which plant they must go to? How does the butterfly, for example, realize which plant it must go to when there are a whole lot of plants? This is very important. How does the bee know where to go and pick its pollen? If you come to the Indian Institute of Science in the month of February and March, you will find the flowers are out in full bloom, and they have all been put in front of the main building, in front of the statue of J.M. Tata. But if you look very carefully, you find that huge thicket of flowers now covered with bees. There will be blue flowers, red flowers, yellow flowers, all flowers of every color. If you watch the bees very carefully, you will find that bees will go to one flower but not to the neighboring flower. Now the question is, how did they know how to go there? Obviously, there must be a chemical attractant. That is what is called a pheromone. And the pheromones are chemicals which are volatile, therefore they go through the air, and you have receptors which can now sense the chemical. Receptors usually sense the gradient of a chemical, so we also sense. Our smell receptors sense smells. Our taste receptors actually are able to taste different substances. We know the difference between salt and sugar. We know when something is very hot. We can sense temperature. We can sense hot material in both senses of the word hot, spicy as well as hot in terms of temperature. All that can be done. And all these chemicals, sometimes here I show you a chemical structure, the kinds of chemical structure which might attract butterflies to a particular plant or attract bees to a particular plant. Then insects also come to plants, and insects are very important. You know, insects are there everywhere. In fact, look at beetles, there will be beetles of all kinds, all shapes, only you have to look closely. You keep some plants there, you'll find one insect or the other. Many years ago, the scientist J.B.S. Haldane, he was a physical chemist, he was a natural scientist, he was just about everything. He remarked that God loves beetles because there are so many kinds of beetles and insects which have been created in nature. Look at this structure. You will find this structure. Now, of course, you may find fewer of these in Basmapudi because there are very few places where you can have them. But if you come to the campus of the Indian Institute of Science or go to somewhat less built-up areas, you will find the termite mounds. Look at the termite mound very carefully. It's a wonderful structure. Next time you go to a termite mound, take a little bit of courage and put your finger into one of those holes that you see there. No snake will come out. But if you put your finger in there, you will find that it's slightly cooler there. It's slightly cooler there because those holes have been made and the structure has been constructed in such a way that the termites have actually understood many of the principles of what today we call green architecture, where you can have a building which is cool without having to waste energy on air conditioning, so that air currents can be drawn in and drawn out in wonderful ways. But how does the termite build this structure? It doesn't build this structure by itself. It needs help. What help does it get? It gets help from this fungus. The fungus is a microorganism. You can't see it. The soil is full of fungi and bacteria. Out of those thousands of types of fungi and bacteria which are present in the soil, the termite picks only one for collaboration. They have a symbiotic relationship in which the fungus farming termite farms a specific fungus. The termite farms a specific fungus, and together they make chemical secretions. They first communicate with one another, then there are chemical secretions, and together they manage to put the structure together. The fungus also provides chemicals which prevent other insects and other termites from coming close and colonizing this particular mound. Therefore, there's an enormous amount of communication. Now today, if all of us got together to keep the neighbors out of National College, we will communicate by sound. We tell one another, go to the gates, close them, don't allow anybody inside. But here, there are chemicals which are secreted. These chemicals would now be repellent chemicals, sometimes toxic chemicals. And all this chemistry is already done in nature. It does not require any human intervention or any human industry at all. The sociobiologist E.O. Wilson, many years ago working at Harvard, called the fungus farming termites one of evolution's master clockwork, tireless, repetitive, and precise, more complicated than any human invention and unimaginably old. E.O. Wilson was the man who coined the word sociobiology. And really, that is a study of the biology of social insects, because insects are social. Bees build colonies, wasps build colonies, they communicate with one another, they have a hierarchy, there are queens, there are workers. When the queen dies, there is a succession plan already in place. Sometimes you will find the bees are probably even better than all our political parties because they already have a succession plan in place. And they don't fight with one another as much as people do. Although you are having a social science forum, I might say that where the social sciences actually intersect with the hard sciences is biology. It is in biology that you will really start seeing the fusion of both the sciences and the social sciences. There is one other subject in which you can sometimes see a little bit of fusion of sciences, particularly mathematics, and the discipline itself, that is economics. Now you will find in economics one branch of applied mathematics, game theory, which is often used. But game theory's most powerful application originally was not in economics, but actually in biology. Therefore, there is a connection between biology and economics. There's also a connection between the study of social behavior in human beings and social behavior in insects. But the social behavior in insects is very strongly controlled by chemicals. To some extent, it may also be there in human beings, but we don't quite know. Look at this example. This is the starting point of chemical ecology. This is an insect which is called the bombardier beetle. This bombardier beetle often has trouble with ants. They are actually the fire ants, which produce formic acid and would like to overcome the insects. Even today you will find if there's a dead cockroach, you'll find ants appear from nowhere and manage to transport it away. If there's a dead insect of some other kind, also they will transport it away because they use it. The bombardier beetle would now like to get rid of these fire ants which are now tormenting it. What does it do? It produces a spray. It produces a spray which is actually almost a flame. It's got hydrogen peroxide. It actually has a temperature of 100 degrees. We all believe that biochemistry can be done at the temperature of the body, which is 37.5 degrees. But biochemistry can also be done at 100 degrees, and that's what happens in this explosive discharge which it makes. How does it make this? It makes this by having these wonderful chemicals that you can see in two chambers and then mixes them to produce hydrogen peroxide. Where are the peroxides used? Today, the next time you get a speaker from the Indian Space Research Organization, you should ask them about peroxides. The peroxides are used as propellants. The peroxides are explosive. and the peroxides are also used by beetles. You can see that when this paper, which appeared in the year, this is 1999, but the original paper is 1969. I like this paper because this is the year that I went to do my PhD. If I had read and understood this paper, maybe I would have become a chemical ecologist well before the subject was known. In fact, in science, if there are any students here who want to do research, I would say it's more important to get into a field which nobody knows anything about. You must only recognize it maybe 2 or 3 decades in advance, then of course you will be there. If you go along with fashion, you are very unlikely really to be able to make a major impact. But this name, Thomas Eisner, is the name that I want to point out because 30 years later, in 1999, when technologies and other things have improved very much, he says, spray aiming in the bombardier beetle, photographic evidence, and continues to publish in this area. Eisner is in fact the man who is the founder of the field of chemical ecology and gave the field the name chemical ecology. Eisner's last student, a young American lady, is now a faculty member in the National Center for Biological Sciences here in Bangalore. And once you should invite her here to come and give you a talk on chemical ecology because she was his last student and she is probably one of the true disciples in taking the field of chemical ecology. Those of you who know me will know that this is not really my field, but is the field in which I got interested because the field involves both chemistry and biology. But over the last year or so, I have been collecting some references because of this program which is starting in Bangalore and also trying to get my engineering friends now interested. You can see that this is a paper in the Journal of Mechanical Engineering Science. This is a paper in the Journal of Power and Energy, 2012-2007. So in the 21st century, there are people now trying to use the lessons learned from the beetles, that is bio-inspired research in the area of mechanical engineering and in the area of power generation. So there's a great deal to be learned in biology. So biology is full of remarkable phenomena. I will show you one here. There is this ladybird beetle. It now carries biological weapons. So if it's there in a given area and it wants to clear the area of all other competitors, it secretes this chemical. It secretes this rather simple chemical. But what does this chemical do? It gets rid of other competing insects. It gets rid of other bacteria and fungi which might sit on the insect and now cause disease in the insect. And therefore the molecule that it produces might also be useful to human beings. Because we would also like to get rid of pests who come into us. So this would be biological pest control. We would also like new antimicrobials. Antibiotics, which Dr. Ramanov talked to me about while we were having coffee. Antimicrobials in the general world. Today, if you get a bacterial infection, you need an antibiotic. If you get a fungal infection, which is even worse, you will need not an antibiotic, but a specific antifungal compound. But all these creatures in nature are also dodging bacteria and fungi. So biological warfare or biological defense and offense gives rise to wonderful chemicals. Chemical technology is vastly sophisticated in biology. You should also remember in the Bangalore Science Forum one more thing. How has in the 20th century human advance in science been propelled? Human advance in science has been largely propelled by, in the West, by the requirement for defense research. Whether it is the discovery of the laser, the discovery of all kinds of radar and radio communication, which is the precursor of today's information technology, all of these have been fueled by huge investments in defense research. Even Alexander Fleming's discovery of penicillin was operationalized with large amounts of penicillin produced. The imperative for that was the Second World War. And penicillin saved more lives at the end of the Second World War than any other invention. But biology is doing the same thing all the time. So we should learn from nature. But Eisner died a few years ago. But in 2003, he said new disciplines arise by a convergence of interest. Chemical ecology is the product of a partnership between biologists and natural products chemists, united by shared vision and empowered by complementary skills. The vision stems from the realization that all organisms emit chemical signals and that all in their respective ways respond to the chemical emissions of others. Even today, there are people who think that even in human interactions, many times these interactions are chemical driven. Sometimes you like someone, sometimes you don't like someone. You believe that you have made a conscious decision. Sometimes your decision might have already been made for you by other substances and creatures who are completely unknown. How many chemicals are there? I've shown this slide before, but I keep updating the slide. You can see the date. Yesterday evening I made this on the 30th of July 2015. I did this for 3 minutes yesterday along with a colleague of mine, and he managed to capture this screen here. You can see how many substances there are. 583 there, it's gone to 609 there. So in 3 minutes so many substances have been added to the chemical abstracts counter. So I think the number of chemicals is growing roughly like India's population, constantly. You can watch their rates over time. I have shown slides like this in lectures over the past few years, and only yesterday when I was making this slide, I was suggesting to my colleague that we go back and look at the counter that we made 5 years ago to see how much things have changed over a period of 5 years. We will immediately know the rate of growth. This certainly, I think, grows at a much faster rate than India's GDP. Biology, and this is a problem that I have been interested in my own laboratory, this is the problem of the cone snail, which is a marine snail. All of you know the shells. In fact, I must say the shells in India are actually, which we work with, the cones, are collected off the Rameshwaram coast. And one of the late President Abdul Kalam's family members actually has a shop right there on the Rameshwaram shore which sells these shells. I think it is even called Kalam shells. And it's a wonderful collection. And the snail now can't move very quickly. Since it can't move very quickly, it has to eat. You know, if you're a tiger, you can sort of get and catch your prey. Of course, if you're a lion and a male lion, you're usually very lazy and you'll just be sitting there. But the female lioness will now go and catch the prey and bring it back. And then the male lion will also feed. But here they can't move. So what they do is they immobilize prey. So if there's a fish or anything else, a worm or something moving in the vicinity, they shoot some poison into it. They have a harpoon-like structure which is coated with a poison which will immobilize. And once the prey is immobilized, they will slowly pull it and eat it as they are doing over here. This problem came to my lab through my colleague, the late Professor K.S. Krishnan at the Tata Institute of Fundamental Research. He passed away last year, unfortunately. But he was, in fact, one of the persons who was a true naturalist. He would go everywhere and he would find wonder in biology. If you brought him to National College, he will immediately find a biological phenomenon right there in the corner of your quadrangle, and you will wonder, look, I have been walking that way so many days, but I never noticed. Unfortunately, he is no longer with us, but he brought this problem to my lab, and really what we have been doing is studying all the molecules which are now present in the venom, which allows this immobilization. But the very same molecules which are used for immobilization might also be the molecules which are useful in human pharmaceuticals, because the kinds of receptor molecules that the snail has, and the kind of receptor and the snail's prey has, and what we have, are very similar, because there is a lot of similarity over evolutionary timescales in biology. This is the apparatus. There's a bulb-like thing, there's a duct in which the chemicals are produced, Then there is harpoon-like structure which is coated, and this is thrown. Those of you who have read the book Moby Dick will know that when whales are hunted, they have these harpoons which they shoot out with a gun and goes and hits the prey. Even today you will find that the forest department people, when they want to immobilize leopards or tigers, every now and then you will find a story that a leopard sort of wandered into some place which is habitated by human beings. They hit it with a dart. That dart which is shot into them contains things which immobilize them and which sometimes anesthetize them completely. All these chemicals come from nature. Bees, for instance, coat the sting with these chemicals, and when they put the sting into you, if you've been stung by a bee, and on the campus of the Indian Institute of Science, you can be very frequently stung by a bee, especially near the director's office. There are two places where you get stung. One is inside the director's house, and the other is the director's office, because they're both in very, very old buildings where big beehives are in fact present in abundance. Sometimes when they sting you, you have this very intense pain. In addition to the very intense pain, you also have a feeling of numbness. Normally you will associate numbness with a lack of pain, but there's such a mixture of chemicals that you actually get both sensations at the same time. So these are wonderful phenomena that one can see, and one can study these molecules. What are the differences in molecules between wasps and bees? Once again, at the GKVK campus, Professor Chandrasekhara and his colleagues are in fact studying these wasps and looking at the chemicals that can come from them. But in analyzing the chemicals that are produced in nature, we are really have to do in the chemistry laboratory the subject of chemical analysis. It is at this point that you will find that chemical analysis becomes very important. There was a report in the Bangalore newspapers that the laboratory for food analysis, which had done the Maggi noodles analysis, there was a press statement which said, we found glutamate, but not monosodium glutamate. All that this statement revealed was that the chemistry of glutamic acid was not known to the person who was making the statement. So chemistry is very important. So before you ban every food in sight, or you ban everything, in fact you will finally realize that you will have to end up banning even human beings altogether if you want your environment completely free of all kinds of difficult chemicals. They are mixtures of substances. They can be the products in a laboratory, or they can be natural extracts. You have to separate them, determine them. This is what chemists do all the time. They use all these techniques which are there, instruments of various kinds, and have also done this. But the nature of the molecules that are investigated sometimes, when you extract the molecules, some molecules will get extracted with water, some molecules will get extracted with organic solvent. You can also do this experiment in the kitchen. If you put a tea bag in, tea that you get is what the hot water extracts. That's the decoction of tea or coffee. But there are lots of other substances which you can extract afterwards by extracting the residue with other kinds of more powerful solvents. You could, for example, pour alcohol. If you are a person who consumes alcohol regularly, then you can in fact pull out a bottle of alcohol, pour it over the residue and ask, what does it extract? So that's how extractions are actually done, and afterwards studied. All the techniques for studying these come from physics, and they pass into chemistry. But I won't worry about it. One technique which I have used a great deal is mass spectrometry, which allows you to weigh the molecule. And since atoms have mass, molecules have a mass which corresponds to the summation of the masses of the atoms. Therefore, molecules can be distinguished on the basis of their masses and on the masses of the fragments that you get when you weigh them. Today, for example, I could distinguish between all of you on the basis of your masses, provided I was able to weigh you accurately enough. If I was able to weigh you to a fraction of a milligram, to a microgram, then I could give you a biometric, which is your mass, and then you can put it onto your Aadhaar card. Except that biometric might change depending upon your age and upon your eating habits as you go along. But you can analyze molecules. Here, for example, is a meteorite which has been analyzed by mass spectrometry. Every line corresponds to a mass. and you can see that there are a total of at least 14,000 different chemical formulae in this, and it's a meteorite sample. Now you can ask the question, where did this meteorite come from? It came from outer space. And since it came from outer space, are some of those molecules extraterrestrial? And this is what people try to study. Looking at the isotope ratios of these molecules, one can in fact ask this question, are those chemicals of extraterrestrial origin or are they of terrestrial origin? They're of great interest. You can study anything. You can study your blood, for example. Two major proteins in your blood. Hemoglobin, which gives you the red cell, the red color, and albumin, which you have. It's sometimes, I think particularly nowadays, very often in hospitals, when you have liver disease, they give you albumin. Hospitals are wonderful. They put up a bottle and put an intravenous feed here, and every patient hopes that the doctor knows what they're putting in. And there's already protocols for this. Albumin is used. You can separate, you poke yourself, get a little blood, centrifuge it, you will get hemoglobin down here, albumin up there, and then of course you can shoot them into mass spectrometers and you can analyze both of them. All this can be done today with today's technologies. But I want to go back to the question of nature and the chemicals that it produces. How does nature produce chemicals? Everybody knows DNA. So the information for everything is programmed into DNA. Now DNA now is used, this is the genetic code, which is in abbreviated form. They're all chemicals, but we just abbreviate them with 4 alphabets, A, T, G, and C. So the genetic code or genomic information is just a string of letters. When today we talk about information technology, I think the future frontier of information technology is big data analysis, which is big data coming from human genome analysis. Your genome and my genome and the next man's genome, they're all different. So if you have 6 or 7 billion human beings on the planet, there are 6 or 7 billion human genomes. Each one of these human genomes contains about 3.5 billion letters. So there's a huge amount of information. If you want to know how many letters, take any book that you have, count the number of letters in one page, multiply by the number of pages, you will get the number of letters in that book. Even if the book this size, you will get a finite number of letters. Now you can think about 3.6 billion letters, you can multiply 3.6 billion by another 7 billion, and then you have a huge number. That's genomic information. But that information is converted into proteins. That is the process of translation of that information. The word translation is literally used. Information encoded in DNA is translated into proteins. What are proteins? Proteins are the molecules which do all the work in your body. They do all the chemistry in your body. They give your body the structure that you have. They give the skin the structure that it has, and so forth. They're very important. But they're also the machines which carry out chemical operations. Therefore, you will find that you have to understand this. So the DNA sequence is a 4-letter alphabet, the protein sequence is a 20-letter alphabet. And then, of course, you need to understand the grammar which you have here, just like a language. That's what biologists today, molecular biologists, are really doing. Eventually, you get a functional, 3-dimensional molecule which carries out the operations that biology needs. Even in the case of the co-stem, how many molecules are there in the toxin? It turns out that one snail probably produces in that toxin, which it is shooting into a fish or into a worm, it produces as many as 1,000 molecules, in excess of 1,000 molecules. Or does this remarkably. Those molecules now, if you were to find out what those 1,000 molecules are, it's a huge problem of analysis. We can ask, can we find the information encoded in the snail genome? One can do that. Today what one can do is one can take the DNA of the snail. Actually what you do really is to take what is called RNA isolated from the snail, convert it into DNA, and then break this DNA into pieces. When you break this into pieces, if you have 1,000,000 letters, you break these 1,000,000 letters into groups of 100 letters. You have millions of pieces now, but you don't know how they are joined. And now you have to assemble them. There are a few simple rules for joining. And these few simple rules are the rules of molecular biology. This is the grammar. But once you have these rules, how do you do this joining when you have millions and millions of pieces? You do it because of algorithms which have been developed by computer scientists today, and these algorithms work very well. I'll give you an analogy here. If you had a big thick book like this, but it had not been bound, it was completely loose, you're carrying it to the binder, and then it falls down. At the same time that it falls down, there's a strong gust of wind which disperses these 1,000 pages all over the quadrangle outside. Now your job is to pick up those 1,000 pieces and put them all back in order. Unfortunately, the author has forgotten to put the page numbers. How would you do this? The only way you would do this, you would have to read the text, and then if you come to the end of this, you'll have to find the next page. But then if you have a 1,000-page book, you have one page, and then you have 199. Now if you were to do this manually, it would take you a substantial amount of time, and undoubtedly you would have to have a computer do it for you. Unfortunately, if you were to have a computer do it for you, you'll have to teach the computer the rules of English grammar now. You'll have to teach it the meaning, and the computer would have to learn. It's not something that the computer can do very easily. On the other hand, the grammar of nucleic acid sequences or DNA is substantially simpler than the grammar of English or Hindi or Kannada or any other human language. There are finally just proteins that must be made, or protein-like molecules. There are two mechanisms. One mechanism which this goes directly from the DNA route. The other is the DNA now makes protein molecules which now take chemicals from the surroundings and join them to get other molecules. Most of the complex molecules of nature are made by non-ribosomal synthesis. So when you look at natural products chemistry, you're talking about molecules made by microorganisms, bacteria, fungi, plants, animals. I don't use viruses, although most of you will be worried about viruses because every fever you get is ascribed to a virus. Viruses have very limited chemistry. Viruses are true parasites. They have no independent existence. This is why viruses, once they have gotten into you, if you manage to survive for a little while, they'll go away. They are self-limiting. On the other hand, not bacteria and fungi, because their chemistry is very complicated. We call these secondary metabolites, the molecules that they produce. But many years ago, a microbiologist wrote the secondary metabolism represents the splendid idiosyncratic diversity of nature, endowing different species with specific solutions to biological problems. Remember, what does Darwinian selection tell you? Every organism adapts and is selected to survive in a given environment. Therefore, every organism evolves depending upon the environment which it is. For those of you who are young, you will remember that there are some portions of your hand which you use much more frequently today than we used to before. One of them is the forefinger, this one. Because you use the mouse this way, or now in all those smartphones and other things, you do this way. You use this finger. You don't use this one, you don't use this one, use this one. It might turn out that after another 1,000 or 2,000 years, you will find that this finger is slightly stronger and the other fingers have become slightly weaker, because that would be evolutionary adaptation on human time scales. Unfortunately, we will not be around to see this adaptation in action. But this adaptation in action, you can see if you look around nature. You will see that all of us, I don't know how many of the older people here have back problems. If you go and investigate, you will find back problems are more common even with the young today. That is because we were never intended to be standing like this. We were actually supposed to be on all fours. The monkeys still don't have that knowledge, but we do. And that is largely because of the evolutionary transition, which has now made us vertical. It has given us some advantages, but it has also given us some disadvantages. So one must have great respect for biological evolution at all scales. Now I will sort of quickly come to something that I must mention in the Science Forum. I've talked about molecules and so forth, but I will make a brief digression. The digression here is because a few days ago, one of Bangalore's most distinguished citizens, a friend of mine, Mr. Narayan, because we were in fact colleagues at the Indian Institute of Technology, Kanpur, in the late 1960s, came to a convocation and then he made this wonderful statement, no invention, earth-shaking idea for India in 60 years. And this is a statement which the press liked, the general public liked, and everybody began to blame the Indian Institute of Science, the Indian Institutes of Technology, and every other institution in India. Now, of course, one must distinguish between some words in science. There's the word discovery, there's the word invention, and there's the word innovation. All of these are used in the context of science. Now, of course, the information technology industry has advanced dramatically in Bangalore. We have Indian Institutes of Information Technology also, which train more people in the area of information technology. Now one can ask, what are the inventions which have come and what are the discoveries that have come from this dramatic advance that India has made? You can ask, have they been innovative? The answer is yes. Innovation means something different. Innovation could be something like the retail store. Innovation is what we see today with every youngster in the age group of 20 to 30 now produces, unfortunately I don't have one, but I'll take my old mobile out and show you. It's not smart, but will produce an app for a smartphone. This will be an application which will tell you how to order food in Basonguri. It will be an application which will tell you how to get an Ola cab. But this is now innovation. It's an innovation which may be a short-lived innovation because it might not last very long. Otherwise, there may be long-lived innovation. The idea of the outsourcing industry is a long-lived innovation. But discovery and invention sometimes means something different. Discovery is something that a laboratory scientist would like to do. Like Fleming, you should be able to discover something by doing something wrong. Fleming did an experiment he should not do, and he didn't really do it. It happened unconsciously, so he made a discovery. Inventions are something that you sometimes do purposefully. Edison discovered the light bulb which has illuminated our lives, but he did it purposefully. Edison could have found the electron, but he did not worry about finding the electron. He was worried about producing a light bulb. Einstein, what did he do? Because you have his picture there at the back. Einstein, everybody knows that Einstein is a great scientist. And now you ask the question, relativity, special, general, whatever it might be. I'm sure theoretical physicists have come here and explained this. Is it a discovery or is it an invention? Now you might have difficulty in a philosophical discussion on what it might be. But I would like to say that relativity, for instance, might be classified really as an invention. It's an invention of the human mind. It's the way you think, the way you think about everything. But the area where discovery has been most fertile, by accident, has been Fleming's discovery of penicillin. And I'm going to take a few minutes here to show you that this is important. Here I've taken some headlines from the newspapers, because I thought the newspapers also are a powerful source of inspiration, whether it's Mr. Murthy or whether it is the problem of antibiotic resistance. Here, for example, the World Health Organization declares antibiotic-resistant bacteria the global threat. New worry, resistance to last antibiotic surfaces in India. And here it says, target ignorance to tackle resistance in chemistry work. And look at this, how this article starts in 2015, after the discovery of the New Delhi beta-lactamases. Here is an organism, a pathogenic organism in human beings, which is resistant to antibiotics. the protein that it produces to break the beta-lactam antibiotics, it's called New Delhi beta-lactamase. Our politicians did not like this when the name first came. The public did not like this. They said, how can you name an organism after the capital of India? And nobody cares. You can shout away as much as you like, but the organism came from New Delhi. Somebody called it New Delhi beta-lactamase. That it will remain so in the scientific literature. It's today called, the gene is called NDM1. What is the abbreviation? It is the abbreviation of India's capital. So I think in the Bangalore Science Forum, you might ask, what has been discovered in India? What has been discovered in India is a drug-resistant organism. Who discovered it? Because a patient took this organism to a hospital somewhere in the West. So these are the beta-lactam antibiotics. Penicillin here, amoxicillin here. Cephalosporin here, and this is the beta-lactam. That's the chemical entity. If you break this chemical entity, that is a beta-lactamase. It's a protein which makes it. Bacteria have genes which make beta-lactamases. They mutate very quickly. If you change the structure, they change the protein and they break it down. Microorganisms are smarter chemists than the chemists working in the pharmaceutical industry. Here, for example, is a molecule which is called a carbapenem, meropenem. A sulfur atom which is present here has been replaced by a carbon atom. That's all. This is the last line defense today against drug-resistant and antibiotics. If today you get a hospital infection, and there are people getting hospital infections in Bangalore regularly, there are people getting hospital infections all over India, you're likely to get a drug-resistant infection. They will treat you with a more and more powerful antibiotics which will create other problems, but they won't create problems to the organism because the organism itself is resistant. Eventually, in many cases of intense drug resistance, there is only one outcome, and that is death. Because the last line antibiotics have failed. That is the one. How do you create drug resistance? You create drug resistance by indiscriminate prescription of antibiotics, because when you kill the organisms which are susceptible, the only organisms which survive are those which are not susceptible. They then multiply. And therefore, by Darwinian selection, you have selected out the drug-resistant organisms. So when doctors prescribe antibiotics indiscriminately, when pharmacists give out antibiotics without prescription, what they are doing is they are doing a great disservice to the general population. There are other antibiotics, extremely toxic, vancomycin and colistin. This is a hugely nephrotoxic molecule, but this is what hospitals administer when all else has failed. But what will happen is, even when these are administered, all else will fail, but only a little bit later. So that's the molecule. I have indicated the beta-lactam. What does the beta-lactamase look like? It's a machine. It's a small molecular machine which looks like this. And it, like a pair of scissors, it will cut the molecule over here. It will do biochemistry on the molecule and break it down. Now there are approaches which are appearing in the literature, and I thought I would show you one. This has just appeared in 2015. This is a new antibiotic which kills pathogens, the drug-resistant pathogens, without detectable resistance so far under laboratory conditions. This is a marvelous molecule, and this is how it looks. This is a control, this is oxacillin, this is the last line antibiotic, vancomycin, this is the new antibiotic, and you can see that this tube is clear. If it's cloudy, it's full of organisms. If it's clear, the organisms have died. How did they find this? They found this by trying to culture in the laboratory organisms which cannot be cultured in the laboratory. Because organisms grow in mud outside, in the soil. So they made a little device which allows them to culture, which they call the eye chip, which allows them to culture organisms in an environment in which they survive. And after that, pull out enough of the molecule to do further studies. Here is the molecule which comes out. It overcomes metallo-beta-lactamase resistance in this. So here, for instance, if you have a resistant organism, and you add this molecule here, which now again comes from a microorganism, it now rescues this. Here is survival percentage of mice which have been infected. At this point, if you add this molecule, the mice survive. If you don't, the mice die. So these are new researches which have come out in 2014 and 2015. These address the problem of overcoming antibiotic resistance. It is this kind of research which really needs to be done very much greater in our surroundings. The first anti-tuberculosis drug followed penicillin. That was streptomycin. This was discovered by Salman Waxman many years ago. And in his 1952 Nobel lecture, he actually quoted from the Christian scriptures. He said, the Lord hath created medicines out of the earth, and he that is wise will not err them. So even in the ancient scriptures, whether they are Indian, Chinese, or Christian, or Western, you will find that this reference to natural medicines has always been there. And this is a microbiologist who found the third set of organisms in the tree of life: bacteria, archaea, eukarya. All the animals, including us, are here. This is the bacteria. And the third tree of life are the archaea. He discovered them. And in a tribute to him on the internet, I found this. It had his picture and this quotation from the old Greek literature, Pliny the Elder, where it says, nature is to be found in her entirety, nowhere more than in her smallest creatures. Those smallest creatures can today be seen only under microscopes. So many of the, although I started with the subject of chemical ecology, you can see that the reason why chemical ecology is likely to be very important in the future is because not only will the understanding of the molecules with which organisms communicate be very important, but we will also from this be able to use these molecules in other ways usefully. So if an organism is producing a molecule to attack a bacterium or a fungus, we might also use the same molecule when we want to attack a bacterium or a fungus which is pathogenic to us. We can learn a great deal from biology, and I believe that in the years to come, chemistry's real future will be to target and find the entire diversity of molecules that happen in biology. Thank you very much.
Good evening, ladies and gentlemen. It's my privilege and honor to thank you, sir, for giving such a wonderful talk on chemical ecology. It has been very informative and interesting, sir. On behalf of Bangalore Science Forum and all of us here, I propose vote of thanks to Dr. P. Balaram. I am happy to announce that we have today completed the 38th Annual Science Festival. I would like to thank you all for your participation and making this event grand success. Thank you very much. Next week there will be a film show on science subject. The Bangalore Social Science Forum program next week, 10th August 2015, Dr. Guru Chandra ಇವರು ಜಾನಪದ ಸಾಹಿತ್ಯದಲ್ಲಿ ಜೀವನ ಮೌಲ್ಯಗಳು ಅದರ ಬಗ್ಗೆ ಮಾತಾಡ್ತಾರೆ. 24th August 2015, Professor S. Joseph will be taking on interior earth movement. All are cordially invited. The session is open for discussion.
Hello, sir. Being a chemist, what is your point or view on medicines like Ayurvedic medicines and English medicine? How are the difference from Ayurvedic medicine and English medicine chemically?
See, Western medicine normally uses pure substances which are chemically analyzed and have also gone through several testing protocols and regulatory procedures. Ayurvedic medicines, on the other hand, do not use single molecules, but they use natural mixtures. And these mixtures themselves are prepared by the Ayurvedic pharmacies by procedures which have come down over the ages. There is a program now in India which has been pioneered by Dr. M.S. Variathan, who is one of India's foremost scholars of Ayurvedic medicine. He has persuaded the government to start a program which is called Ayurvedic Biology, where both Ayurvedic practitioners and modern scientists from the best institutions in India together have research projects funded to understand the nature. But there's a very, very clear difference. This is why even if you take formulations which are made by companies which export, like Dabur and Himalaya and all, in the West, they are all sold as food supplements. They are not sold as pharmaceuticals. On the other hand, in India sometimes, you will get all those also in the pharmacies that we have. Then you will get them in the supermarket. So there is a philosophical difference between, as far as science is concerned.
Excuse me, sir. So my question is, what is that this thing, insect which emits light? What is the chemical combination in that time?
The firefly.
Yeah, yeah.
Yes. The firefly is very, very interesting. There what it does is, it has an organic molecule which is a very high energy molecule. It is on twisted straight, it wants to break. At the moment that it wants to do this, from another place it produces an enzyme which now breaks this, and the energy that is there is now, the molecule raises to an excited molecular level, and then when it falls back to the ground state, it emits photons. See, like when we absorb, we'll go to when we absorb light, we'll go to a higher state. If you're able to create a molecule in the excited state by a chemical process, and then allow it to come down, it will emit light. So that's the process which is called bioluminescence. You can do this even in the laboratory in a process called chemiluminescence. It's a fascinating thing to find. Actually, in the institute, in the house where I used to live, after it had rained and all dark, only trees, you could just find all over the place.
Yes. Sir, just my next question is, if somebody want to poison some people or animal, I think, in food, how it is going to be sensed? You were telling about the sensing of some material, you know. If somebody want to put some poison in some edible things, yeah, how is it going to be analyzed, detected by animal or man?
So you are not going to detect it very easily. On the other hand, that is why we have food regulatory people. Your food analysis laboratories and forensic analysis laboratories must have a very high degree of chemical analysis. Unfortunately, in India, they don't have a very high degree of chemical analysis. But it's not enough to spend money. You must also have people.
If somebody present you with anonymity, somebody presents some food just to kill me or some
if somebody has malignant intention, you can't smell it.
Sir, viruses being the parasites, and you said that they cannot remain independently. But how come this HIV person who is infected, he becomes a carrier for life?
What happens with HIV is this: once you have HIV infection often does not mean that you have the symptoms of AIDS as a disease. You have HIV infection. When full-blown disease is there, it is detected. Today, even anti-HIV agents which are used, which are antivirals, azidothioprine and so forth, which they give, they reduce the viral load. Whether they very often they reduce the viral load very substantially. So people do not necessarily have to be infective throughout life. But there is a problem here, because even tuberculosis, which is not a viral disease but a bacterial disease, every one of us in this room is probably TB positive. Because Indian populations are by and large TB positive because they've been exposed to TB. It doesn't mean that we are diseased. It means that we have microorganisms which are sitting inside cells in the body macrophages, they are doing nothing, they are staying there. You will sometimes find in India a very interesting thing. As we live older and older, sometimes a disease that you have forgotten comes back because your immune system slowly gets depressed and then these organisms begin to grow. There are many such things. TB lies dormant. Viruses also sometimes.
Sir, thank you for this great lecture. I had a question on snail venom, which we were talking about. Is it produced during the time it preys, or is it produced and stored earlier? If it is stored earlier, how does it prevent from the snail getting paralyzed itself?
So, in fact, that is done by all these organisms. Even bacteria which produce molecules which kill other bacteria, they have mechanisms by which synthesis and secretion into the outside environment are done very well. In the case of the snail, the answer to your question is we don't know. Because there's no doubt that the composition of the venom changes over time. But that you can study only if you keep snails in an aquarium in the laboratory and then look at the venom they produce when you put a fish there and look at the venom that they are producing as what you would call a steady state. Such experiments are actually being done in the United States by a person who keeps the snails in an aquarium and then milks them. There also the environmental stimulus is there.
Okay. So thank you for a wonderful lecture. My question is, how far is the action of pheromones in biological pest control, sir?
Very much. One can hope that biological pest control, as generally talked about today, is less by use of molecules produced by biology rather by using whole organisms. Like for example, there are organisms like fungus like Trichoderma, Viridae and so forth, which people are using. So biological pest control usually uses whole organisms and they may produce molecules which then are used. But even plants, the area, in fact chemical ecology which is starting in NCBS has one more young faculty member who's joined, a young lady who's joined there. They all come from the field of plant pathology. And plants sometimes coexist with endosymbionts which are either bacteria or fungi. They live along with them because they give them defense against other insects which might cause a problem.
Thank you.
We heard a wonderful lecture, sir. Can you elaborate on the fact that how does monosodium glutamate is used in neurotransmitter? Just said about that. Is it glutamic acid or sodium glutamate?
So what I want to tell you here really was that there is no distinction. Because if I put glutamic acid now into water, I just put it into distilled water, it will break into the ion glutamate and H+. If I put monosodium glutamate into water, it will break into the ion glutamate and sodium ions. What is actually functional is the glutamate ion. And therefore, whatever you do, you get the glutamate ion. And therefore, it is that which is functioning as a neurotransmitter. So monosodium glutamate might be excitatory in something extra.
I'm going to ask you the same question, and you said that the chemist who said that there was glutamic acid in what he was talking about. You didn't use those exact words, but that's what you said. Can you expand on that a little bit?
That is because when he detected glutamic acid, it's equivalent to detecting monosodium glutamate. And as far as any activity it may or may not have in somebody who consumes it. On the other hand, I'm not very sure because I only saw this in the newspaper report. I do not even know what kind of analysis which was done. Because you see, if you're going to detect glutamic acid, any food that you have, and especially if it is a cooked food or it has been a processed food, you will detect glutamate. This is why glutamate level is important. How much of glutamate? And this is why even in America it is the level which is important. Here what has happened is that there's been a public scare. This is why even the companies have gone to court. And eventually I suspect if they get powerful enough testimony from experts, the case will fall apart.
Whatever is
levels of lead which were considered significant, of water being considered significant.
Pardon?
The levels of lead in the food, in the noodles, which were possibly more important than the presence or absence of lead.
Well, that I don't know, because I think this presence of lead is sometimes also a red herring. Because you then test all your food and test all your water for lead and see what are the results that you get. The amount of lead that we must be consuming is pretty substantial. Because lead comes really through the pipes that we have. It's not added deliberately.